Okay, so I'm going to start going over our genetics practice problems here. So most of you have already done this first one, but I'm going to go over it anyways. Very basic problem. It's probably as basic as it gets. First step for these problems is I tend to underline all the biology words. In humans, the allele for albinism is recessive to the allele for normal skin pigmentation. So I'll circle normal and albinism here. So albinism is recessive. If the albinism is recessive, that means that our normal allele is dominant. Okay, if here's another biology word right here, heterozygotes. Two heterozygotes have children. What is the chance that a child will have normal skin pigment? What is the chance that they will be albino? So we want to know the chance of albino and the chance of normal skin. So number one I always do for these types of problems is I make a key. Okay, so let's see, albinism is recessive, normal, and I'm going to use A's since albinism starts with A, big A, little a. Albinism is a recessive disorder, so I'm going to label this little a as albinism. And I will label this big A as normal. Step 2, parent genotypes. Well, we want to know what genotype these parents have. Of course, a genotype is made up of two alleles, and both of the parents, it looks like, according to the question, are heterozygous, which means parent 1 is going to be big A, little a. Parent 2 is also going to be big A, little a. Remember, I'll put this out in parentheses over here. Heterozygous is different. Hetero means different, which means you have a different initial allele to the second allele. So big A, little a, big A, little a, dominant A, recessive A, dominant A, recessive A. These are the parent genotypes. Okay. Now, especially in these monohybrid crosses, next thing we got to do is we got to cross these two. Okay, so if one parent, I'm going to put one parent up top. Just put big A and little a. I'm going to draw a little sperm around here so you can visualize that these are gametes that we're going to be fertilizing here. And then on the other side, big A and little a. Okay. This part I think is pretty self-explanatory. We're taking down and across. Big A comes down, big A comes across, give you big A, little a. Here we have, of course, always keep your dominant allele first. Big A, little a, big A, little a, and finally little a, little a. If we populate, I'll put arrows indicating where these are coming from. If we populate all the way down. So, normal pigment child. Well, first thing you got to ask yourself is which ones are normal. I'll put in green. I'll go ahead and put the normal up here. This one's normal because all you have to be normal is at least one dominant allele. Does this have at least one dominant allele? Yes, it has two. These heterozygotes both have one dominant allele, which means they are also normal. I'll switch. So we have 3 that are normal. So I'm just, out of our 4 possible children, 3 out of 4 are normal. Or you can also put 75%. Okay, let's look at albino here. Pretty self-explanatory probably from this point on. I'll do it in blue here. Here's albino. Of course, 1 out of 4 is albino. We know they're albino because in order to show the albinism trait, you have to have both lowercase or both recessive alleles. That's the only way they're shown. Again, the dominants always cover up that recessive. But since we have 2 recessives here, we're going to have 1 out of 4 are going to be albino, or you could also put 25%. Okay, let me scroll down just a little bit here. So we've got another question here. If the child is normal, this is the key phrase right here. If the child is normal, so we're assuming the child is normal, what is the chance that it is a carrier or heterozygous, right? A carrier is a heterozygous individual, somebody who doesn't show the trait, but they carry the allele for the trait. So, a carrier for the albino allele. So, if it says if the child is normal, that means we're looking at these three. I'll put them in red. Here's a normal, put some stars next to the normal ones. All right, if the child is normal. So, we're assuming the child is normal. What is the chance that they're heterozygous? I'll go ahead and, let's see, I'm running out of colors here and do this beige, weird beige color right here. Okay, these are the normal children. How many of these are heterozygous? Well, this one and this one, which would be 2 out of 3. So 2, I'll put that in a better color here. 2 out of 3, or 66.67%, something like that. Okay, so that's number 1. I'm going to go ahead and move on to number 2. Okay. In purple people eaters, kind of a made-up animal here, one horn is dominant and no horns is recessive. So I'm going to underline dominant and I'm going to underline recessive here because those are biology words. And it clearly defines what's dominant and what's recessive. Pretty simple here. Here's some phenotypes: no horns and one horn. So I'm going to circle these. One horn is dominant, no horns is recessive. Show the cross of a purple people eater that is heterozygous for horns with a purple people eater that does not have horns. Okay, summarize the genotypes. That's the first thing we're going to summarize, and the phenotypes of the possible offspring. Period right there. Okay. Well, first things first. Number 1, we got to make a key. Make a key. Okay. One horn is dominant, so I'm going to use H, since we're talking about horns here. So big H, since it's dominant, is going to mean one horn. One horn. Ah, that's terrible. One horn. Okay, little h is going to equal no horn. No horn. Okay, now we have a key. Step 2. Parent genotypes. What are the parent genotypes? Well, we know a genotype is made up of 2 alleles. The first genotype we need to know, at least in this case, they will be made up before later, but we'll get to that later. In this case, we have a heterozygous for horns as our first parent, right? There's a purple people eater that is heterozygous for horns. So if something is heterozygous, remember the first and second allele are different, crossed with a purple people eater that does not have horns. No horns. If a purple people eater does not have horns, that means that by definition it has to be little h, because that's the only way a purple people eater can have no horns, is to have no dominant allele. This is the only way. So now we have the parents' genotypes. Step 3, let's cross them. All right, I'm going to put a big H and a little h up top. I'm going to put a little h and a little h on the side. And then, of course, at this point, it's just crossing them down. So when it says to summarize the genotypes and phenotypes, what that means is I want you to tell me essentially what is the percentage or fraction or ratio of each of these, right? So if we look at it, let's look at the genotypes first. Let's look at the first genotype we have here. We have big H, little h, right? We have 2 out of 4. This is 1, 2, 3, 4. 2 of these out of 4. So for big H, little h, it's going to be 50%. Or you can also put 1/2. That would be fine. Fine with me. Let's do little h now. I'm just going to do it off here to the side. Little h is equal to Also, 2, if you'll notice there's 2 little h squares out of a total of 4 squares. So this is also 50% or 1/2, however you want to put it. Now it also asks you to summarize the phenotypes, which in some cases these can be very different than the genotypes ratios. So phenotypes, remember, physical traits, pH, right? Physical traits. First trait we have is one horn, which is dominant. I'm going to put one horn here is equal to, and again, we're looking at any genotype that has a dominant allele, of which there's 2, 1, 2 out of 4, right? Which is going to be 1/2. Again, 50% are one horn, or about 1/2. Now let's look at the no horn. This is the recessive genotype here. We have little h, we have 2 of them out of a total of 4 squared. That's also going to be 50% or 1/2. Okay, I think that's it for the purple people leaders. Let me scroll down and let's do number 3. Okay. In humans, the brown eye allele is dominant to the blue eye allele. If 2 heterozygotes mate, what will be the likely genotype and phenotype ratios of the offspring? So, brown eye allele is dominant, so I'm gonna circle brown eye here. And I'm just gonna point to dominant. That lets me know, hey, brown eyes is dominant to the blue eye allele. Now it doesn't say the word recessive, but if brown eye is dominant, that means that blue eye kind of has to be recessive. So I'm just gonna denote that out here to the side to help me out. If 2 heterozygotes, there's another biology word, heterozygotes, mate, what will be the likely, and here's a couple more biology words, genotype and phenotype ratios of the offspring. Remember, phenotypes, physical traits, genotypes are those 2 4-letters. Something that makes up an individual's genetics in relation to a certain trait. Okay, step 1. Probably starting to get sick of this, but it is worth it to go through every step, every time. First step is to make a key. Well, brown eyes is dominant, so I'm going to go ahead and put a big B, meaning brown eyes. Blue eyes is recessive. Little b is going to mean blue eyes. Okay, number 2, parent genotypes. I'm just going to put GTs, parent genotypes. Well, it says if 2 heterozygotes mate, remember that prefix hetero typically means different, which means that both of them are going to have different alleles. So you're going to have a big B and a little b crossed with a big B and a little b. So it says if 2 heterozygotes mate. Okay, now we have parent genotypes. Now let's cross them. Okay, put a big B and a little b. Over here I'm going to put a big B and a little b. Okay, now you guys know how to do this part, I think. Big B, big B, big B, little b, big B, little b, little b, little b. Okay, what will be the likely genotype and phenotype ratios of the offspring? So ratios are kind of one we haven't dealt with yet, but something you have to be able to recognize. You probably remember them from math. But I'll go ahead and list this out. First thing I want to look at is the genotypes here. There's 3 different possible genotypes. Big B, big B, big B, little b, and little b, little b. Now, all I have to do to find a ratio is to count up how many I have of each and put a colon between them. So if I count big B's, put a semicolon here, there's 1 total big B or homozygous dominant genotype in here. So I'm going to put a 1 with a colon. All right, now let's count our big B, little b's. There's 2 big B, little b's. So I'm quite simply going to put a 2 in there and another colon. And then little b, homozygous recessive down here. And we're going to put a 1 because I only have 1 of those. Okay, this is one of those situations where our genotype and phenotype ratios might be a little bit different because I only have 2 phenotypes, brown eyes or blue eyes. So let's go, I'll go in this order. We're going to do similar to what we did up here. Brown eyes and blue eyes are our two possible phenotypes. Well, let's count up our brown eyes first. 1, 2, 3. 3, I'm going to put a colon there. And then let's count up our blue eyes. There's 1 down here, so I'm just going to put 1. Okay, you now have calculated the phenotype and genotype ratios of the offspring of this pairing. Okay, I'm going to stop this here, then I'm going to move to the other page and we'll work over those problems. Thank you for listening.